All right. So we're going to start on page 9, and we haven't taken the notes yet, but we are going to go and use the summary space for a video that we're going to watch. So please, on the top line where it says summary type, write the title Video 5 Facts About DNA, and then number from 1 to 5 in that summary space. While you're watching the video, please make sure you are writing down 5 facts about DNA. If you need to pause the video, if you need to go back, that's fine. Just make sure that you are able to get 5 good facts. All right, here you go.
DNA, also known as deoxyribonucleic acid, is a molecule. It's a bunch of atoms stuck together. In the case of DNA, these atoms combine to form the shape of a long, spiraling ladder, sort of like this one here. If you ever studied biology or saw the movie Jurassic Park, you probably heard that DNA acts as a blueprint or a recipe for a living thing. But how? How on earth can a mere molecule act as a blueprint for something as complex and wonderful as a tree, a dog, or a dinosaur? To help answer that question, let's first take a quick look at amino acids. Amino acids are tiny little chemicals inside our bodies that are so important, they're often referred to as the building blocks of life. There's about 20 different kinds, each with their own unique shape. The neat thing about them is they can be attached to each other kind of like Legos to produce an endless variety of larger particles known as proteins. Amino acids make up proteins. Proteins, along with other chemicals, combine to form living cells. Cells make up tissues. Tissues make up organs. And organs, when they're all put together and functioning, of course, combine to form living creatures like you and me. These proteins that make up our bodies, and keep in mind there's millions of different kinds of proteins, they each have to be formed in the perfect shape in order to function. If they're the wrong shape, they usually won't work. That's where DNA comes in. DNA does a lot of interesting things, some of which we don't fully understand, but one of its main and most well understood functions is to tell amino acids how to line up and form themselves into the perfect protein shapes. In theory, if the right proteins are built at the right time and in the right place, everything else from cells to organs to entire creatures will come out just fine. This here is a simplified model of DNA. It shows us that the steps of the ladder are made up of 4 different kinds of chemicals shown here by different colors and letters. If you look at just one half of the molecule, you can read its chemical sequence or genetic code from top to bottom, sort of like a book. A single strand of DNA is extremely long, millions of letters long. It spends most of its life coiled up like a noodle, living inside the nucleus or the centerpiece of a cell. Amino acids, however, live outside the nucleus in what's called the cytoplasm. To help DNA interact with the cytoplasm and convert those amino acids into proteins, special chemicals inside the nucleus make partial copies of the DNA code. These partial copies, called RNA, look a lot like DNA, but they're shorter, of course, and they're missing one of their sides. Their small shape and size allows them to fit through tiny pores in the nucleus, out to the cytoplasm, and into the mouth of another particle called a ribosome. Ribosomes are protein-building machines. They read the RNA code 3 letters at a time, suck amino acids out of their surroundings, and stick them together in a chain according to the RNA code. As the chain grows, it bends, it folds, and it sticks to itself to form a perfectly shaped protein. Every 3 letters of the RNA code tell the ribosome which of the 20 different kinds of amino acids should be added next. For example, CAA tells the ribosome to grab a glutamine. AGU tells it to grab a serine, and so on. Once a protein is built, it can then go on to do a number of different things, one of which could be to help form a brand new cell. So to answer the original question, what is DNA? DNA is a molecular blueprint for a living thing. How does it work? DNA creates RNA, RNA creates protein, proteins go on to form life. This entire process, as complicated, as sophisticated, as magical as it might seem, is entirely based in chemistry. It can be studied, it can be understood.
All right, before you move on, look to see that you have 5 good solid facts about DNA. And if you don't, please go back and rewatch the video until you do. All right, now we'll move on to the notes. DNA stands for deoxyribonucleic acid. And that sounds like a big, long, kind of scary word, but if you look at the different parts, you're going to find word parts that you recognize that tell us a lot about it. Ribo tells us about protein. Remember, ribosomes make protein. Nucleic, it's found in the nucleus. Oxy, there's oxygen in this molecule. DNA is the genetic code in all cells. This is basically the instructions for how you become you. During cell division, that DNA is copied and passed on to the new cells. And we get those identical copies, those cloned cells, through the process of mitosis. To copy itself, the DNA goes through a process that we call unzipping. And that's because it totally looks like a zipper that is being unzipped. It makes a copy of the other half as it separates so that you end up with 2 identical strands of DNA. Now, this DNA shape is this unique shape called a double helix. That's the name of this shape that looks like someone took a ladder and sort of twisted it around. The rungs of the ladder always pair in a really specific way. So if you look at your diagram, you'll see these names of these 4, they're called bases: adenine, thymine, guanine, and cytosine. Now, you're not expected to know those names. I don't expect you to remember them, but I do expect you to remember their first letter and who's a partner with who. A is always a partner for T, and C is always a partner for G. And if you look right now at the diagram, you will notice that that is true. We'll come back to this diagram in a few minutes.
DNA is a 2-stranded molecule, twisted to form a double helix. Inside, you can see the bases, like rungs in a ladder. During DNA replication, the molecule unzips. New bases float in from the surroundings and are bonded into place. The pairing is always the same. Adenine pairs up with thymine, cytosine pairs up with guanine. Because of base pairing, the 2 new strands are identical.
So sometimes with all this copying and unzipping, a mistake is made. And those mistakes in the copying of the DNA are called mutations. Now, when you hear mutation, you probably think of weird things, right? Like maybe you think of something like this, or maybe even something like this. Okay, just kidding. But not all...
Mutation is when a gene is damaged, for example, by radiation. In a point mutation, one base pair is replaced by another. Now that the coding has changed, the gene may not work properly. Some mutations affect whole chromosomes. In a deletion, a segment of the chromosome becomes detached and is lost. Sometimes the segment turns round, completely changing the order of the genes.
So mutations, you think of right away something crazy or weird, but actually a lot of mutations, we don't even know they happen to us. So mutations can be harmful, they can be actually helpful, or in some cases they just have no effect on the organism when we don't even know they've happened. Now sometimes when they're harmful, these can be the kinds of things that keep the organism from growing, repairing, or maintaining itself. Okay, so now what you're going to do is grab a highlighter and take a minute to highlight the key words. And as you finish with highlighting, you can go ahead and put your highlighter right back into the basket and move on to writing your 3 questions. pause your video for a second right now while you write these 3 questions. Remember to use 3 different question starters. All right, so now let's do something with this diagram. You can use any 4 colors. You do not need to use the colors that I've used, but you're going to use 4 different colors to add some color to your diagram. So choose your first color, and you're going to color code the word adenine, and then go over to the diagram and find anywhere that there is the letter A and color in that part of the ladder that has the letter A. Take a second color and do the same thing for thymine, for guanine, and for cytosine.